All right, so you should be ready to go right now.
Okay. All right, welcome everybody to this exciting seminar.
I'm not going to tell you.
I am Simone Baumann-Pickering, and I am a scientist at the Scripps Institution of Oceanography. I lead the acoustic ecology lab here. And I'll be talking about studying marine mammals with sound. So at UCSD, we have an overarching theme that is called Understanding and Protecting the Planet. Now, if you look at a map of our beautiful planet, then you can see that there's a lot of blue, as you're all probably very well aware. And turns out that about 90% of our Earth's livable volume is actually in the deep ocean. And that means anything in a biological sense deeper than 200 meters water depth. There's, as in many other places of our Earth, a number of threats, namely such as climate change, pollution, man-made structures that are being installed, and extraction of living, as well as non-living resources, such as, well, living being all the fisheries, and non-living, such as oil exploration. And all of that is going to impact the deep ocean. However, we understand at this point relatively little about the affected ecosystems, and the nature of these impacts remain somewhat unknown. And so in the crux of things is that the vast majority of our ocean essentially remains unseen and unquantified. And I'll be looking at that in the context of this lecture with the conservation of marine mammals inhabiting these deep ocean regions. So to look at marine mammals and conservation, all marine mammal species, to start out with, are protected under the Marine Mammal Protection Act, instated 1972. Now, some of those marine mammals are also protected under the Endangered Species Act, which was instated 1973. Now, there's a beautiful web page installed by the International Union for the Conservation of Nature, or short IUCN. It's called the Red List of Threatened Species. And if you type in iucnredlist.org, you come to the starting page. And you can type in any which species name you want to, and that can be not only animals, but also all plants and fungus and whatever not else. And so my example species, one of my favorites that I have been working on that I typed into this is Cuvier's beaked whale. So if we do that, then you come to the starting page on all the information that people have gathered on Cuvier's beaked whales. So one thing I'd like to point out to start out with is this category down here. So Cuvier's beaked whales are considered as least concern, so nothing major to worry about for the most part. But there are other categories such as then near threatened, vulnerable, endangered, critically endangered, and if it gets real bad, then extinct in the wild and extinct. But then there's the other side, the gray boxes, where you have animals where there's just not enough data to say anything for sure about how they're doing, so they're data deficient, and they're not evaluated. All right, so what all goes into this to come to a categorization? So if you were to scroll down on that page, you would find entries on the geographic distribution of the species, usually then with a range map. In this case here, you can see the geographic range of Cuvier's beaked whales with the orange areas of the world. And for all and everything, in order to understand whether a species is vulnerable or threatened or endangered, you're going to need to know how many of them are there. So the population numbers. And not only the population number itself, but then also the trend. How are they doing over time? Is it stable, or is it decreasing, or is it increasing? Then in order to come up with some numbers for population trends and status, you also need information about life history and habitat and ecology. Things like how old do they get? How often do they reproduce? How many babies do they have when they reproduce? How long do they take a break between babies? What kind of area do they prefer? Deep ocean, shallow ocean? In this case, we know it's a Cuvier's beaked whale. They like deep ocean. What do they eat? Who do they hang out with? What kind of ocean conditions do they favor? And so on. So that's the life history, habitat, ecology type of aspect. So then, in order to say anything about trends, you'll also need to know the threats. I mentioned a few earlier already. The big topics in general are climate change, pollution, disease, but then also fishing. And in this case, fishing can be related to bycatch. How often are they being caught or entangled in fishing nets, for example? But also, how does fishing impact their resources that they rely on, and so on. And then if it is a species of concern, then usually there's a list of conservation actions that are already being undertaken that are on this webpage. All right, so now in looking at marine mammals as a whole, there's about 89 species that I'll be focusing on of whales, dolphins, and porpoises called cetaceans. And of those, 2 of them are critically endangered, namely the vaquita here and the Atlantic humpback dolphin. The vaquita of those is probably the most critically endangered, as there are at this point likely only about 10 or less than 10 adult animals left. And all the conservation actions have not been particularly successful. Now, there's another 10 that are listed as endangered. You can see over on this side. Some of them actually have success stories, such as these 2, sei whale and blue whale. After stop of whaling, these species have gradually increased in overall numbers. But the rest of them, you can see that they have decreasing trends, and then some are just so inaccessible that their status is unknown. Now, if you do the numbers here, if you count up all those that are endangered, or critically endangered and vulnerable, you come up to about a fifth of all these species that have some serious problems. Then you have 26 species that are actually considered data deficient, so where we don't even know much about them at all. And of those, there's about 20 that are in that large family of beaked whales, where the Cuvier's beaked whale is my favorite example belongs to. So now that's about a third of them being data deficient. So if you add that all together, we only have half of all cetacean species that are doing well, whereas the other half is either of concern or status unknown. So we have a lot of catching up to do. So now I would like to focus on what we can do with our technology using acoustic methods to contribute towards filling up all the information relevant for this large data deficient group of beaked whales. So all of the beaked whales undergo very long, very deep dives. So they're far offshore, hardly ever encountered, and they breathe a few times at the surface, then they go for a long dive that can be a record holder, over 2 hours of a dive. And then they go down as deep as 3,000 meters. That was a tagged animal, and that was the longest recorded yet, and the deepest. So a lot of these species that are considered data deficient in this family actually have never been seen alive in the field, or if they've been seen, they've not been able to identify them as such. So many of them we only know because a couple of few individuals stranded on a beach somewhere, and we were then able to say, oh yeah, that's a new species. And it turns out, though, that the two most common ones, namely here Blainville's beaked whales shown in the graph, and then the Cuvier's beaked whales that I mentioned earlier, they have stranded in relation to Navy sonar exercises, where there's anti-submarine warfare exercises. And these animals react very strongly to these sounds and strand dead on the beach. And the Navy has been sued to pursue research to understand better why this happens and how it happens. And so money has gone into researching these data deficient species, and we've learned a lot over the past decade or so. And one thing that we have learned, and that's where the passive acoustics come in, is that they can be differentiated to the species level. And that is what we'll be looking at later on. And so that's very convenient to be able to monitor these rarely seen and potentially not rarely occurring species. So let's look at the different technologies available to us to do that type of work. On the right, you can see what we in this lab use a lot is these autonomous seafloor recorders. You drop them in the ocean, they stay there for a few months, up to a year or even longer, and collect acoustic data continuously. And then you pick them back up to bring them home and look at what happened during that time period. Alternative is you have a cabled array that is the graph up here. It's a long cable on the seafloor, different nodes, many instruments attached to it. And it brings in the data in real time while you sit at your computer in the office. And then another opportunity for these autonomous systems are gliders. So some have a surface float and something dangling off of them and then drive around in the ocean, and others are underneath, like that glider to the left. And that kind of zigzags up and down the water column and collects data on the way. So then alternatively, if you don't go with these somewhat autonomous systems, you can do surveys on a ship. So one thing is that here, for example, we deploy a profiling float that is also an autonomous system, but it has to be observed more closely. So it kind of floats up and down the water column too, and after a few days, you go pick it up. Or you have here a towed array that is being towed behind the ship on a long wire and sends the signal through the wire into the lab, and then we can record the signals and look what we see while we record. This is usually done for toothed whales, so higher frequency signals. For low frequency baleen whale signals, we have so-called sonar buoys. That's a Navy instrument that we're allowed to use. And that radio transmits the signal. The ship, you drop it in, the ship drives away, and the radio transmits to the lab. And while you're doing that, you then have visual observers looking out for animals at sea and try to identify who's there and what kind of sounds do we hear. And lastly, another option is tagging. And tagging does not necessarily have to be with acoustic sensors, but it can be. And so up top here, you can see, for example, a satellite location tag, and they transmit the position of the animals as they move around. It stays on it for multiple weeks to months. Whereas down here, these are shorter term tags. On the left, it has audio and video and some auxiliary data. And to the right here, it's an acoustic tag. And those are suction cup attachment. They stick them on with a long pole, and it records for a few hours to maybe a couple days. So now I'm segueing a little bit and not focusing exclusively on the marine mammal side of things, but as we record these acoustic data, we also record a bunch of other sounds, and that is relevant in a broader context of understanding the environment the animals are swimming in. So we collect data from the earth, from animals, but then also from people making sounds in the ocean. So as you look at geological sounds, you can see here earthquakes, hydrothermal vents, methane seeps, ice, lightning, rain, waves. And I've brought an example of a thunderstorm. So you hear the rain and you hear the thunder rolling underwater. Sounds pretty much like it sounds above water. And then you have the biology. So now we're going to the marine mammal side. Here's an example of a dolphin whistling and clicking. But not only marine mammal make sound, but also fish and other species like crustacean, that is shrimp, but also animals grazing on rocks like sea urchins are making sound. So let's hear some fish. And then here, that was a croaker. And here we have snapping shrimp. And if you're snorkeling, you're most likely to hear this crackling in the background in warmer waters especially. And so that is snapping shrimp. Let's listen to that. And that segues actually right into the next topic, being the anthropogenic sound. So you heard the crackling, snapping shrimp along, but then at some point there was a motor that was put into gear, and then you could hear the revving up of some engine, and a small boat was driving away. And at that point, you didn't hear anything of the snapping shrimp, but all you heard was the sound of that boat going by. So there's a lot of anthropogenic noise, which is a big topic in the acoustic realm. All the impact of noise on these animals, and not only marine mammals, that's the topic of today, but any marine animal, truly. And people sound, that includes so-called air guns from seismic surveys looking for oil. Echo sounders. Most any ship is equipped with some sort of sounder that emits a signal, looks at backscatter, so the echo that's coming back, and that can be Navy, oceanographic research, fishing vessels looking for fish, oil exploration looking for the sea floor. But then there's also pile driving, so that's essentially where there's big piles being driven into the ground to install wind turbines, or again for oil exploration. And then you have recreational things like jet skis that are being driven around a bay. The big ship sounds, container ships are continuously driving on our oceans, but also recreational activities like small fishing vessels. And then we hear explosions. And explosion, that's kind of an odd thing in that it's not only our navies blowing things up to test, but also fishermen use these small firecrackers called seal bombs to deter, as a deterrent, a sound deterrent device for seals and sea lions to keep out of their nets. So here I brought another small example of a jet ski, and you all are probably aware of how that's going to sound like, having been at the beach. So there's abundance of these types of sounds out there. So now what do we do with that? Well, you can imagine that if you walk along the ocean, or if you walk downtown, whatever city it is that you're living in, it's going to be sounding quite a bit different. And the same is true when you put these sensors into different habitats. And so you're going to be able to know something about weather, about climate, about who's living there in terms of biodiversity, what are these animals doing behaviorally, and so on and so forth. So now we're going to the marine mammal side of things. So here we see a cartoon of a sperm whale and a Cuvier's beaked whale clicking. So they emit these echolocation sounds that are hitting, in this case, their favorite prey, a squid, and that sound is bouncing back and they're interpreting the echo and then go forage and search for that. And in the meantime, here, our sensor is sitting conspicuously on the seafloor and listening into what these animals are doing. So now we have echolocation clicks of sperm whales up here. So that's a very regular metronomic sound. And they keep doing that on and on and on. And the way we look at it, though, is in these spectrogram pictures where we have the frequency here on the y-axis and the time on the x-axis. In this case, it's 40-minute stretch. And you have sperm whales in the first 20 minutes, and then it changes, and here you have beaked whales. Now, as you realize, we just listened to sperm whales, and they're relatively low in frequency. People actually cannot hear anything above that yellow line. And so we don't hear beaked whale clicks. That's why we have to have these kind of visuals to then see them for our analysis purposes. So we have to first, as we take the data that comes in from the ocean, understand who is calling, in this case, echolocating. And this is an overview of all, not all, a good number of beaked whales in the Pacific. And the way we're looking at it, we're looking at the amplitude over time. That's the first line here. Then below that is the frequency over time, and the energy of that signal is in the darker black it is, the more energy there is. And then down here you have a amplitude over the frequency. So it's kind of a cross section of the one in the center. And then the last thing we're looking at is the metronomic rate that we listen to for the sperm whale. So that's that number that is in the time series box up here. So it says 200 for Baird's beaked whale, 360 for Longman's beaked whale. And so they are relatively clear in the regular, they're consistent in their echolocation patterns. And so with this, we can figure out who's who. And now you can see that some of those that I now marked green are associated already to a certain species, while others, those that are marked yellow, we don't know who's producing those. And so the type of approaches we do is then looking at biogeography. So where do they occur geographically? Based on strandings and visual sightings, these blue lines have been, these blue maps have been made by others. And on top of that, we now marked in red where we have heard certain signals. And just by overlaying that, we kind of now have an idea who is likely going to be producing our midstream ones. So for example, this one down here, the parent beaked whale, that is a species that we only know from, I think, 4 dead animals that have washed ashore along the Southern California coast. So now we think we have a good candidate for what kind of signal they're producing, and we're learning a lot about this particular signal that way. So then I had a question earlier in the Q&A section. How do we know to estimate the population of these animals? So for one, you're doing that usually visually, but in our case, these animals are hardly ever seen. So we approach this to do that acoustically. And let me move to the next slide. The question always is, how many animals are there, right? And for it to do that acoustically, you would have to count how many clicks you have in a certain time period, let's say 5 minutes. And then you have to know how often would you expect there to be a click. So that comes where our click rate comes in. So if it's metronomic, there's a certain rate at which they're clicking. But then sometimes they don't do that, they never do that continuously. They kind of take breaks, right? They go to the surface, they breathe a little, and then they go dive again and click again. And so you have to know how long those breaks are. And not only that, but you have to imagine that a click only travels so far before you can't hear it anymore. So you have to know how loud is the click that is being emitted and how is it going to travel through the water column to your sensor. And additionally, the animals have kind of like a flashlight beam. So they don't just broadcast in all directions, but they look and point their sonar beam to a certain direction, kind of like a flashlight, if you shine a flashlight out. And so you have a directional beam. And with all these, you can then estimate what the probability is of detecting a click and what the probability is of the animal clicking. And with that, you can then calculate a density around your sensor. And this is an example that we calculated 2 different sites and then the progression over time. And so at this one side, we roughly had 7 animals on average in 1,000 square kilometer area around the sensor. And in the other one, we had about 2 animals. And so in a normal scenario, to get population estimates, you have a ship that travels through and there's a density estimation method that then counts, how far out do we see, what's the probability of seeing that animal, and so forth. So now after that, once we have the numbers straight, there's the matter of the whole life history thing. And so in this case, I'm going to be showing something on what is their feeding behavior. Cuvier's beaked whales, like I mentioned earlier, same as sperm whales, like to eat deep sea squid. We know that from stomach content analyses. Now, deep sea squid isn't a very easy thing to measure because they're really good at avoiding nets and they're not really visible just from observations at the sea surface. So when you look at, hold on, I gotta click here. So we did an experiment where we put a whole bunch of different instruments at the seafloor using not only a single sensor, passive acoustics, but the whole network of sensors where we then can kind of pinpoint by triangulating between the sensors where the location is of the whale as it is swimming through the water column. Additionally, we had echo sounders sitting at the seafloor looking up. And so they were emitting sounds to collect the backscatter energy, like a typical fish finder on a fishing vessel, to understand where animals are in the water column. So we're looking for the squid. Additionally, we took measurements of this is a small CTD, we took measurements of conductivity, temperature, and depth, so salinity and temperature at certain depths. And then we had what's called an acoustic current Doppler, ADCP, acoustic Doppler current profiler. So an instrument that looks for currents, flow of water around that system. And so let's look at the prey side of things first. How can we measure prey? And this is now a view of the backscatter from roughly 900 to 1300 meter water depth over the course of 1 month. And the color in this case indicates how much energy is being scattered back to our recording device. And so you can see that there's a somewhat persistent layer here between roughly 1050 meters and 1150 meters over the entire time. It fluctuates a little bit, but it's mostly there. And then there's kind of this fuzzy layer underneath that. And when we look at that more closely, then you can see this is just 12 hours on that May 1st. There's hardly anything above here. And the reason for that being, that's the so-called oxygen minimum zone. So that's an area that is oxygen restricted, and so nothing can truly live in that region. So it's accumulating below where the oxygen slowly increases again. And then you have this fuzzy layer, and underneath though, you have larger single targets. And so we did some net tows, stratified net tows, where you just do slices within certain water depths. And oops, wrong direction. And we found out that this layer here is largely comprised of zooplankton. So it has a lot of jellies, but also crustaceans in it. Whereas the layer below that has mostly squid and fish, and mostly squid, truly. And so we then asked, well, are the beaked whales actually, because initially that's what I thought, that the beaked whales would be having a field day foraging right through that layer here. But when we looked at the actual dives of them, then we came up with this plot here. So here you have 4 different dives on 4 different days, each about 20 minutes long. And the pink area here, that's where that zooplankton layer is. And that red line shows you the diving beaked whale depth. And down here you see the seafloor. That gray layer around the red one kind of shows uncertainty, where our calculations weren't converging perfectly on a spot. But what strikes me now is that they largely forage in that area where we have these single targets, likely their preferred squid, whereas they only sometimes go into those layers where the zooplankton is, and probably there's the squid have to eat something. So I imagine that squid are going to exploiting the zooplankton, and from time to time, the beaked whales are going to just dive into that as well. And I thought you might be interested in seeing how such a dive looks like in 3D. So here is a little video. So you see our sensors, our acoustic sensors here, from which we calculated the track. And so the red is the track, and then these gray lines at the seafloor and on the sides here, they indicate just a projection so that you have a reference in space. Otherwise, it's kind of hard to interpret. So you can see how that beaked whale is just kind of going up and down and meandering around, doing sometimes some pretty fast ascents and descents to capture their prey items. So now we've learned something about where they go and how they find their prey. In general, though, we know now that they are not uniformly distributed, but they like certain habitats. And when you look at this plot here, to the right where that says B, you see our sensors in the Southern California Bight, where we've collected acoustic data over many, many years. And the distribution in the red, the larger the red circles, the more acoustic detections we have at those sensors. And when you look at the oceanography, and that's the plot to the left, then you can see that the California current, which is a cold water current coming down along the coast, bringing cold Arctic water south, you can see that that almost perfectly overlaps with the preferred beaked whale sites that we've identified. Now, the California current is not always looking the same over the course of a year, but it's stronger. It has a stronger dominance kind of in the winter, spring months, and then the summer has influence from flows coming northward and from the south to the north, pushing warmer waters into the region. And then the California current comes back in. And so we see a seasonality in Cuvier's beaked whale presence in the Southern California Bight, and that has been not known at all. And we still don't quite 100% understand why there is a seasonality at all. But you can see how it's kind of stable from January through June, and then it kind of drops off July, September. The animals are more and more absent, and then they come back to their previous numbers. And it has always been the assumption that it has to do with the strength of the California current. And now we were able to, like I said earlier, put some sensors in to measure temperature, and not temperature at the sea surface, but temperature at the depth that the beaked whales are foraging at, where it actually matters for their prey. And so this is a plot now of a whole year of data from, I guess, what is that, October to October. And you can see the temperature and salinity plot at the bottom here over the time period. And at the top, you can see how many detections of beaked whales we had at that particular site. And the temperature and salinity, essentially, a certain water mass has certain properties. And so the combination of temperature, salinity is going to tell you, is it California current water or is it some other kind of water? And in this case, you can see how very rapidly, end of May, early June, there's a complete switch here from kind of this constant looking water mass to a flip where it's all of a sudden getting warmer and lower salinity. And that's the point when all of a sudden these Cuvier's beaked whale disappear. And obviously they don't probably care about this little bit of a temperature change or salinity change, but their prey might. And so the idea is, and we haven't measured that properly, the idea is that as this new water comes in, it brings a complete set of new, a complete new community of animals that go with that water mass and turns over what was there previously. And then at some point it goes back to where it was. And that's then when the animals slowly show up again. So last point that I would like to touch on is what threats do they face? And I mentioned earlier that the initial funding in this topic area was through naval conflict with anthropogenic sounds produced by the Navy, mainly so-called mid-frequency active sonar. And it's not only beaked whales, but it's also blue whales that respond strongly to these sonar sounds. And offshore California here, we have a large naval range where anti-submarine warfare exercises are being carried out. And so we record a lot of this kind of sound. And first of all, we have to, in order to understand whether and what kind of impact that has, we have to quantify how much sonar is there and does it overlap with animal presence. And you'll be surprised to, let me move to that slide. You'll be surprised to see how much sonar there truly is in an area such as Southern California. So this is data from 2009 to 2012 at one particular site. The gray area in the background is nighttime. These kind of bluish shades in between is when we didn't have data. You have the time of day on the x-axis and the dates on the y-axis. These red lines are when sonar was detected, and the blue dots, the small spots, are when Cuvier's beaked whales were detected. Now, if you zoom into that here to the right, you can see a shorter period of time. There are some short periods of sonar use, but then there's also some larger exercises where a long period of sonar use occurs. And so then the major question is, can we acoustically document behavioral impact, and how does that all work? So what we did is we computed a statistical framework. And if we would first focus on the left side, and I'll introduce you how to read these. So essentially what you're asking is, what's the probability of there being a detection of a Cuvier's beaked whale given a certain parameter? And so the higher on the y-axis you are, the higher probability of detecting that Cuvier's beaked whale. The blue stuff at the bottom of these plots shows you the distribution of data that went into that model. So let's go look at the non-sonar variables first. So in this case, the variables that were relevant to explain the patterns in our data was the year. So there's interannual variability, and that probably just has to do with how good of a year is it for these animals to be present to begin with at that particular site. And then it's the time of the day of year. So that's our seasonality. We kind of covered that already, right? So you have the dip kind of in the July through September time period. And then it's also oftentimes that there is some difference in daily activity. In this case, it seems like midday, they vocalize or echolocate more than not. But that is a very variable thing from site to site. So then we're looking at the sonar related variables. So essentially we've taken out all the natural variability with these non-sonar variables, and now we're getting to the meat of it. So the first thing we're looking at is how much time has it been since the last use of sonar? And you can see here that if sonar is being used back to back to back, which is actually a lot of times looking at the blue distribution, then the probability of detecting a Cuvier's beaked whale is relatively low. After about 7 days of nothing, you kind of come to this shoulder here where the animals are using the area pretty consistently. And then we come to an area where we have very, very little data. And that is exactly the 6 years over Christmas. There's been every time over Christmas, New Year, the Navy just takes some time off, and there's a stretch of 3 weeks where nothing happens. And that's when we all of a sudden see a major influx. Now that can be a model error, or it can be actually true. And that would only be seen as we gather more and more data. It could mean that if we really leave that place alone, then the animals truly access that. Equally now, the proportion of sonar per minute essentially means if the more in a minute is covered with sonar, the more annoying it becomes and the less probable it is you're going to see an animal. And equally, the louder the sounds are, the less likely you're going to have an animal there. And the Navy is always going to trying to figure out, well, how loud is already showing a behavioral response. And usually you take a 50% level where you say at 50% you have a response. So this is so-called dose response curve, if you will. And so roughly about, say, 118 dB or so, which is not very loud, truly, you already see a response as a decline of presence. All right, so summing up, because I'm getting here to the end and I want to answer some questions. I think I've shown you quite a few ways how acoustics can help in filling up these data deficiencies and to help in conservation and management of marine mammals. And it's by no means everything that we can do, but there's only so much time that I can spend talking about it. So we can document geographic distribution. We can help identify populations and stocks. We can help estimate density and abundance. We can quantify prey and predator-prey interactions. We understand something about noise and behavioral response through these acoustic methods, and then so much more. All right, so I see that I have a few questions. And let me see if I can answer those. The question is, how did you manage to capture all these sounds, and how long did it take to record them? So I think that was a question related to all the different beaked whale species themselves. And for some of them, we were in the field and we had hydrophones in the water while other people's were looking. And then you can bring that together. Others where we were in an area of the world where this one species just dominates the region. And so if there's one signal that dominates in that, then you can put that together. So similar to those unknown signal plots that I showed you. And how long did it take? I guess, I mean, we're still working on it, and I'm working on it since, I guess, a good 15 years now. All right, I think that one's answered. Then the question is, does climate change affect the California current? And absolutely, yes. So there's a lot of work done at Scripps. There's actually two major programs that look at that kind of thing. It's the so-called Cal Coffee program, which is I'm always butchering that. Let's see if I can get it straight. The California Cooperative Fisheries Investigations. I'm sure that I didn't get that right, but it's Cal Coffee. Anyway, so it's a big program that goes out quarterly. We do measurements of the physical properties of the water. We measure plankton, fish eggs. We look for cetaceans. We look for seabirds. We do underway measurements and station measurements, so to get down into the deep. So that's and then there's another program that's called the California Current Ecosystem Long-Term Ecological Research, so the CCE-LTER. And together, and this program looks more about dynamics and functions. And in short, there's lots of changes already observed, such as changes in species that are present in the Southern California Bight now, changes in strength of upwelling, changes in water temperatures overall. We have had heat wave after heat wave in the North Pacific. Changes in the timing of things, so so-called phenology changes, and so forth. Okay, I think I've answered this one. Then the question is here, would you say the Navy then has an overall positive or negative effect on these animals? Overall, that's a difficult question to ask, right? So the question ultimately is, on a species and global population level, my guess is no, because they're not globally active such that you would expect a complete wipeout of the species due to naval exercises. On a local level, I would venture to say very much so. So then it becomes the question, how do you define your management stock? That means, are the animals living in Southern California actively changing genetic material with animals from other regions? And if the population in California were to go down, does that impact only California or other regions as well? Also, if the population were to decline, which is still unknown at this point, truly, what kind of side effects would that have? How are they integrated into the overall food chain and the ecosystem? To, if they were taken out of the equation, what would that mean? So it's a non-trivial question that I can't answer definitively one way or another. Okay, so I have no more questions online. I don't know if anybody else would like to know something. I'm not sure. There's another question. Okay. How do other marine animals like coral reefs affect the more active populations like fish and crustaceans and their sonar? I'm not 100% sure that I do understand fully, but so there's a lot of talk about masking, and you have a certain sound that you want to transmit to another individual. Yeah, if you want to communicate, you want to transmit your sound from a sender to another, to a receiver, right? So I want to find a mate as a fish. I'm sitting in a coral reef and I want to find my mate. And so then the question is, what kind of active space do I have under natural conditions, and what changes once the sound properties, the noise level changes, right? How much smaller is my active radius that I can communicate over? And so that can be that actually the coral reefs are pretty noisy, and animals themselves are pretty noisy. The weather, you know, if you have a big storm, that's noisy. And so animals have adapted to how they change their signals with noise. They can get louder, they can repeat themselves so that they make sure maybe the first sound didn't go through, but the second and third did. They can change their frequency, so they can shift a little bit higher to get out of the noise span that is impacting them, and so forth. but that comes at a cost. Oftentimes, so say if you have to call a whole lot more, that's costly, or if you have to scream to be louder, that's costly. And it's always the question in terms of how can we document how these kinds of noises are impacting the animals on their success to reproduce, because that's ultimately what it comes down to, right? In a population sense. Do I get enough food to make a living? And then does that give me enough energy to reproduce, to keep my species going? And optimally increasing, because then it's prosperous and it's doing well. And so that is a difficult thing to measure. But from people, we know that noise can change your hormonal levels. It can be due to increased stress, and so your overall well-being might be impacted. You could be more receptive to diseases because you're kind of weak to begin with, and so on. So there's lots to be said about the stress of noise, especially if it's noise that it's not naturally occurring in your environment, that you know is safe, but that is a scary kind of noise that you don't know what to do with. I don't know if I answered the question for that, but that's as much as I have for this. We have a couple more minutes, so if anybody wants to ask another question, now's the time. So one thing, I'll just babble a little bit, that we've been doing lately is a lot of those experiments where we look at many different data streams coming together and trying to understand how oceanography is changing prey presence and the type of prey that is present, and then how these different marine mammal predators react to that. So that's a big ongoing topic. Another big ongoing topic is definitely how do we automatically work through these data. There's so much data that we collect. We've probably now got about 2 petabytes or so of data, and more than 2 petabytes of data. And you can manually work through that. So we have computational problems to solve to automatically screen through that. Oh, there's a chat window too. There's a question. Would you say marine animals use echolocation but a different species are in some ways able to communicate with each other? Oh, I would definitely say so. Yes, they can listen into each other for sure. All right, well, thank you all for listening, and I hope you'll enjoy the next talk as well.
Thank you so much for joining us.
I heard down the volume.
Yeah, we heard it on our end too. Yeah.
All right.
Thank you again so much.
Yeah, I didn't see the chat window until later. Otherwise, I would have been able to react.
Oh no, you were fine.
All right. And it's weird to but just talking.
Yeah.
All right.
Great. So we're going to start our next one soon, and thank you again.
Thank you. All right, bye.